Finding fossilized behavior in physiology sounds like an impossible feat given how challenging it can be to find actual fossilized parts of these organisms' bodies. At least, the parts have physical substance like bones and teeth. It turns out that behavior in physiology, at least traces of them, can be fossilized. Trace fossils can be footprints, skin impressions, nests, burrows, bite marks, claw marks, even fossilized poop called coprolites. That's all trace fossils. Linking traces with the species that made them can be challenging, but sometimes matching the trace to the trace maker can be fairly obvious. Very rarely do we find body fossils and their associated traces together, in situ, which is Latin for in place, as it was preserved in the ground. The conditions required to preserve trace fossils like tracks and impressions can be more forgiving in some ways, especially chemically. Footprints and nest structures are not going to dissolve in acidic conditions like bones would, but such traces do not survive transport either, but instead just wash away. So, different kinds of fossils and trace fossils can have their own strengths and weaknesses when it comes to increasing their likelihood of preservation. Taphonomy is about looking at how fossils were preserved. What conditions did the organism perish under? What happened to its corpse from the point of death to the point of its final burial? And everything else that may occur between then and the point some lucky fossil hunter finds it. Taphonomy looks beyond the body fossil itself for clues in the matrix surrounding fossils to piece together how they arrived as deep time travelers from the past in our world today. That is not to say that it ignores body fossils. There can be traces on body fossils, like bite or claw marks on bones, or if the specimen was at least partially mummified, there could be skin impressions preserved on the skeleton in its surrounding matrix. There could be pathological bones if the dinosaur suffered an injury and that injury partially healed. Such clues are useful in determining how a fossil organism may have died. In the rocks surrounding a fossil, though, we can find out what happened after an organism died, sometimes. If the specimen happens to be a dinosaur buried in marine sediments, for example, then we would assume that some significant transportation happened between this animal dying in or near a terrestrial environment and the time it was subsequently washed out to sea. Usually, we have to look a little harder to figure out what happened, though, as the bloat and float method of carcass transportation is pretty obvious. Taphonomy also studies how fossils are positioned in situ, and what other fossils are immediately associated with them. If there are a large number of individuals, all of the same species, and that is the only species present, that is a rather large clue. Monospecific or single-species bone beds are usually indicative of some sort of interesting catastrophic event that took a portion of a population quickly. A mixed assemblage helps us understand the composition of an ecosystem. If multiple species are buried together, there's a good chance that they live together in the same environment. It is in this way that taphonomy and the study of trace fossils, ichnology, can help us detect clues that imply behavior in physiology. For example, the clue that a monospecific assemblage representing dozens or more individuals implies that these animals died together and therefore, this species probably lived together in large groups. In a way, this is also how we implied from skull structure that dinosaurs were amniotes, somewhere between crocodiles and birds physiologically. So we could guess based on that relationship that since both bracketing taxa laid eggs, that dinosaurs also laid eggs. We could not rule out the possibility that they retained eggs, though, not until we actually found some dinosaur eggs and or nests. I mentioned earlier in the course that we've known of fossil dinosaur eggs since the 1920s. The first were found in China by the American Museum of Natural History expedition led by Roy Chapman Andrews. It was obvious at this point that at least some dinosaurs were oviparous. meaning they hatched from eggs. There is so much more, though, that we can learn from studying dinosaur eggs more closely. And I'm not just talking about dinosaur embryos fossilized inside these eggs, but the eggshell itself is full of information, even without a nest or even a whole egg. Just from a piece of shell, we can determine if the egg was adapted to being laid in a buried nest or was one that was exposed to open air. Eggs are self-contained life support systems. They have everything a developing embryo needs, except for a place to dump carbon dioxide and to store oxygen. For this reason, eggshell has to be gas permeable, so carbon dioxide can get out and oxygen can get in. The structure and concentration of the pores that facilitate this gas exchange can tell us whether the egg was adapted to being in a buried nest or one that was exposed to open air. Eggs that were buried tend to have larger diameter pores and more of them per area. Eggs that were buried in nests also tended to be rugose or bumpy. The relief between the bumps helped create spaces where the matrix burying the eggs was not in direct contact with the shell surface and increase the surface area of the shell. This is important since the stuff the egg is buried in is another barrier to efficient gas exchange. If you find these eggs together in a nest, they will often be tilted or vertically oriented, which possibly helped with gas exchange by preventing pores from being waterlogged across the whole egg at once if the buried nest got dampened. Vertically oriented eggs in a nest are only possible if the eggs were at least partially buried. Also, from looking at the pore structure and the crystalline structure of the rest of the shell, we can see patterns indicative of evolutionary relationships. The microstructure of eggshell is an inheritable trait and different across groups of different reptiles. Even different groups of dinosaurs have different eggshell microstructures. Studying such microscopic fossil detail is possible using histological techniques, such as thin sectioning slices of shell or other tissues and preparing them for scanning electron microscopy. Histology is the study of tissues and the cells that form those tissues. Contrary to what you may have learned about fossilization, the original material of a bone, tooth, and even fragile stuff like eggshell is almost always still present in its fossilized form. Fossilization does not often involve the complete dissolution of the once living tissue, replacing it with another sediment and precipitates. The vacuities of a fossil may get filled in, and will likely have other minerals precipitate in the fossil, but this is occurring on and around the original calcium phosphate matrix of the bone or tooth. In many cases, the original tissue can still be studied using the same sorts of techniques we study living tissues with. We can even see details at fine subcellular resolution. Unfortunately, we cannot see things as fine as chromosomes through these histological techniques. DNA is a fickle thing, and it would seemingly take a miracle to find significantly long strands, even as much as 1,000,000 years old, let alone 100 million years old. Not that we sequence DNA through a microscope anyway. That is handled by making lots and lots of copies of it, and then chopping them up with enzymes and seeing how they titrate out. Even if we could find dinosaur DNA and a significant enough links to identify genes on it, we would still be very, very far from growing a dinosaur from it. So don't hold your breath waiting for Jurassic Park. It's not happening anytime soon, if ever. Histology can potentially tell us how many birthdays a dinosaur has had, though. Thanks to the seasonal nature of the planet, many trees and animals experience fluctuations in their growth rates. Dinosaur bones sometimes have growth rings that can be counted like those in trees. Periods of optimal growth produce less dense, broader rings where the bone was expanding and growing faster, and new blood vessels invaded these regions of growing bone. Periods of slow growth are marked by lines of higher density rings which developed on an annual basis with the seasons and are unvascularized, meaning no new blood vessels were developing to support growing bone at these times. The annual cyclicality of the rings lends them their name, so they are called annuli. In some instances, bone growth appears to have halted altogether rather than just slowed, and these occurrences are called lags or lines of arrested growth. They could mark sudden or harsher winters, droughts, or some other stress, preventing sufficient nutrition to support growth. Being able to see the relative thickness of optimal growth periods and lags, or annuli, allows us to estimate the growth rates of dinosaurs throughout their lives. Growth rates can have implications on whether a dinosaur was warm or cold-blooded. Warm-blooded or endothermic animals have growth that is hormonally controlled, so they have periods of rapid growth early in life, and then they cease growing once adulthood is achieved. Cold-blooded or ectothermic animals tend to grow constantly from the time they are conceived to the time they die. Whether dinosaurs were ectothermic or endothermic organisms has huge implications on the rest of their physiology and behavior. Since dinosaurs have been found on every continent, as far north and as far south as land was exposed in the Cretaceous, it seems presumable that at least some dinosaurs were endothermic enough to survive these relatively cooler climates. Having a more refined idea of the physiology of dinosaurs is important if we want to estimate ecological relationships, as well as climatological adaptability. Warm-bloodedness requires more calories to support the background metabolism it entails. We would need to estimate that warm-blooded dinosaurs ate more of whatever it was they were eating to survive. Having better knowledge of how much food dinosaurs needed can help us predict population and herd sizes and other facets of their interactions with other organisms. With all we know now, determining whether a dinosaur was a carnivore or herbivore is about as firmly as we can deduce most of their diets. Some traces, like stomach stones, or gastroliths as they're called, are linked to herbivorous diets. In the rare event that we can find stomach contents, that obviously makes things simpler. Coprolites, or fecal matter, can preserve parts of the food that went into making them, but then we have to guess which dinosaur made the coprolite. Teeth are usually what we look to for determining if a dinosaur was a carnivore or not. Meat-eating dinosaurs pretty much all have teeth that look the same. They are conical to blade-like, slightly curved backwards with cutting edges called carini. These edges are usually lined with fine serrations to help them cut flesh more efficiently. Collectively, this suite of features is called zyphodonty, or a zyphodont dentition. Plant-eating dinosaurs have a greater variety of teeth because some are adapted to chewing their food much more than others. Some have spoon or leaf-shaped teeth like iguanas today. Others have relatively simple peg-shaped teeth. In herbivorous dinosaurs that chewed their food, these teeth can be worked quite flat on their occlusal surfaces, where they contact with the teeth in the jaw above or below them in the process of processing plant material.